So what we're interested in is, as it says in the title, precision medicine and global health. When we say precision medicine, what we mean is we try to identify, if you will, groups of neurobehavioral symptoms on the basis of sort of shared biological behavior, biological mechanisms. So this is one aspect, and we're hoping that, of course, in time, models like that will help to at least enrich, if not to sort of transform psychiatric diagnostics. That's one aspect. But the other aspect is, in order for us to sort of really identify those shared biological mechanisms, we of course have to take account of the environment and how environment might shape brain. And environments, as we all know, are very different, for example, in France and in India. So if we want to do precision medicine, and we want as many people as possible to benefit from this, we need to adapt it to sort of local conditions, and this is where the global part comes in. Now, the backbone of the study, or of our work, is the Imogen study, which some of you might know. The Imogen study is going on for 10 years now, even more than 10 years. And the basic design of the study is that we recruited in 8 sites across Europe, including in Paris, 2,000 adolescents at age 14. And we recruited them in order to characterize the biological basis of reinforcement-related behavior. Reinforcement-related behavior in our Operationalisierung, so to switch into German, means reward processing, means impulsiveness, and means emotional processing. We assessed those behavioral domains using structural MRI, task-based functional MRI, as well as DTI, but we also did extensive neuropsychological and behavioral characterizations, as well as functional and genomic and epigenetic studies. So that's the background. And now, importantly, sort of starting as a cross-sectional study, Imogen has come into the years, so to speak, and we have since carried out several follow-ups at age 16 and age 19, and now currently we're completing the follow-up at age 23 years. So I think that we can say we're probably the most comprehensively characterized and largest longitudinal cohort worldwide that captures at least a good extent of adolescent brain development. So here again, to looking at the three sort of constructs we are particularly interested in. I mentioned, we're looking at impulsivity. Here in particular, we're looking at inhibitory control, and we're using a neuroimaging task called the stop signal reaction time task. To measure emotional reactivity, or to measure brain activity during emotional processing. This is a passive viewing task where one looks at angry faces or neutral faces or a control condition. And to measure reward processing, we're using the monetary incentive delay task, essentially a task where money is being promised or made a possibility, and then brain activity is measured in the anticipation of a possible monetary reward, or also if you're looking at the feedback phase into the reaction, the activity of the brain, if you either received or did not receive the money. We will focus on anticipation in this case. So why did we do this? Why do we think these behavioral domains are interesting? I'm telling you, just taking the example of reward processing. And so if you're looking at reward processing, it becomes quite clear that different, so to speak, dysfunctional ways of reward processing are related to very different psychiatric diagnoses. right? And yes, they do include, for example, bipolar disorders or depression and addiction. So it's at least a hypothetical possibility that there are shared underlying biological mechanisms between the two disorders. But of course, if we recourse to typical diagnostic characterization, we will not be able to detect those shared underlying biological bases, simply because the criteria that we use are not based on biological mechanisms. Now, we were in order to sort of approach, so to speak, this problem, and this is very much an approach, right? This is not a ready-made product. This is not something you can apply, but this is sort of just trying to sort of generate additional knowledge and additional understanding to eventually be in a position to sort of translate this understanding into markers that enable stratification and markers that enable prediction of disorder. Which one? Yeah. Oh, okay. So to approach this, we did many things. One of the things we did is first we looked at the genomic architecture of brain heritability. So if you're positing the idea of, sort of biological phenotypes, it's also important to kind of look at the stability of these phenotypes. And one way of looking at the stability and perhaps identify particular risk and resilience factors is to look at genetics. And this is work that was done by Roberto Toro with Thomas Bourgeron here at the Institut Pasteur in Paris. And essentially, it shows you that across different brain regions, you find heritability of about 50%, and this is SNP-based heritability, so this is the kind of heritability you can measure using whole genome association studies, and that this degree of heritability is comparable to heritability of height, heritability of IQ. So, there is a degree of heritability in brain structure, not gigantic, but also not negligible. This here is another interesting work. This was done by Jonas Ricciardi at the time at Stanford. And what they did is they were asking a slightly different question. So they say, okay, we know that genetic variations, genes, are correlated with brain structure and, as a matter of fact, also with brain function. But what we don't know is, what is the actual activity of the genes, the expression of the genes? To what extent does gene expression influence brain activity? That is their question. So what they found is that if you're using some sort of advanced statistical techniques which are based on independent component analysis, then you sort of identify about 139 genes or expressed genes, gene expression profiles in different brain regions, and that these 139 gene expression profiles really kind of seem to be correlated, and you could probably say drive the resting state activity of different resting state networks. Yeah? So now we know two things, we know first that genetic variation correlates with structural and also functional brain features. And we also know that gene expression, the activation of the genes, is actually driving and is influencing brain activity. These are very, very basic kind of findings, right? And remember I told you we're not going to have a product, we're having an approximation, and I think these are the first steps towards this approximation. Now this is here a slightly different situation. You remember that we were interested in reward processing, we were interested in impulsiveness, we were interested in emotional processing. We were interested in these three sort of behavioral domains because we felt, or because it's known actually, that that sort of dysfunctions in each of these domains are correlated with psychopathology, right? So if you're very impulsive, you're probably going to drink more alcohol, and you're going to have perhaps ADHD features, right? If your reward processing is really kind of subdued, you might have negative symptoms as you have in schizophrenia, or you might be depressed, right? And if your emotional processing is dysfunctional, you might end up with an affective or an anxiety disorder. So what we were interested now is we were using these 3 tasks, which are limited to sort of measure these behavioral domains. But we nonetheless used those 3 tasks and looked at what is happening in the brain, which are the areas that these tasks are activating in the brain, and to what extent are these areas distinct, and to what extent are these areas shared across the tasks. And this is just to give you an overview here. So we find, So we find some areas that are distinct for the tasks, but we also find a surprising degree of overlap between the tasks. And that is not only an overlap that is, sort of driven because this is a visual presentation, right? Like you'd have activation in the optical, optical brain regions, in the occipital brain regions. This also sort of involve areas that are in subcortical and cortical brain regions. And it seems, and the slide doesn't show that, but it seems that the actual sort of physical localization of the brain areas is really not the sort of discriminating kind of factor between those 2 behavioral domains. It's much rather sort of network configurations, how these different brain areas are linked up in each of the tasks that do confer the difference. But now, if you're doing this, I mean, this can become interesting potentially, right? Because it might offer you the possibility to sort of link this brain activity with psychopathological symptoms, and then looking, if you're looking at these psychological symptoms across the 3 tasks, you might end up with a signature, right, for each of the psychopathological symptoms that you're investigating. And this is sort of what we try to do here. And again, it's a simple thing, right? It's the strengths and difficulties questionnaire. And here, these are ADHD symptoms overall. This is hyperactivity, this is impulsiveness, this is inattention. Blue is monetary incentive delay, so that's reward processing. The cyan is the impulsiveness, and the red is the emotional processing. And I can't and don't want to go into the details, but what you're seeing here is that in each of these different behavioral domains, you get different activations, both with respect to localization, but also to proportion of any given task. Yeah, so in one word, you're getting different signatures. Yeah? And this is described here. Here again, ADHD, hyperactivity, impulsiveness, inattention, and then actually this is oppositional defiance and conduct disorder. And you see, that the signatures of each of these conditions is distinct. So in a way, we would be able to differentiate those conditions on the basis of the brain signatures. Yeah. Now there is a problem with that. In a way, so far so good, nice and all. But the problem is that when we're looking at this here, and we're looking at the amount of variance that is explained by these particular fingerprints, it's really not. It's a rather small amount of variance that we're explaining. So this is a little bit of a problem. And I think the more we sort of thought about this, the more it became evident to me that this is a conceptual problem. And the conceptual problem arises from the fact that you are essentially having one guy speaking French and the other guy speaking English, right? And say the psychologists speak French and the brain people speak English. And then you sort of bring these 2 sets of data together and try to get, so to speak, a common, some common information out of them. And that sort of limits, you're just going to have a very limited scope And I think, so the problem that you're having here is you're setting up your neuroimaging analyses on the basis of voxel-wise or region of interest-wise statistical analyses. You're analyzing them within the context of neuroimaging, and then you're taking these results, and then you're applying them to something where behavior has been, sort of characterized in the context of, some psychological theories, and then you're sort of praying that they match up, and they do so only to a very limited degree. So we have a problem. Oh, gosh, I'm gonna have to, we're gonna have to do some maneuvering here. No, you know what? Just remember this sort of linguistic problem that we're facing in neurobiological research for a couple of slides. I just want to show you something in between. This is actually quite simple. So we were asking, because we had these we were interested in these brain features and how they relate to psychopathology. We were interested in does binge, well, we know that there is a correlation between binge drinking in adolescence and structural brain in adolescence. So we looked at, and yes, in our sample, we could also replicate this correlation, and we found an accelerated brain volume reduction between 14 and 19 years is associated with increase of binge drinking in frontal and temporal cortices. So far, so good. Other people have found that too. The question that we have is actually, is it the binge drinking that affects the adolescent brain development? And in order to, is the accelerated brain volume reduction observed in binge drinking caused by alcohol is the question, right? And in order to address this question, we relied on what they call like a Bayesian causal analysis. And to make the long story short, no, to our surprise, we thought, great, now we're going to find a toxic effect of moderate binge drinking on adolescent brain development. No. So if you're looking at this causality analysis, you find that yes, binge drinking at age 14, is highly correlated with increase in binge drinking between 14 and 19. But if you're looking at the directionality of the relation of brain volume change and binge drinking and increase in binge drinking, you see that the brain volume change in the majority of cases that is causing actually the changes in binge drinking. So that was interesting. We also then looked at personality factors, not only brain structural factors that might drive drive this the increase in binge drinking. And one of them that we found was impulsivity. Also, it's been known. So again, we find binge drinking at baseline is, causes increase of binge drinking between 14 and 19. But then it's not the change in impulsivity during your adolescent years between 14 and 19 that influences your binge drinking change. In fact, it's your impulsivity at baseline that influences the binge drinking change. So this is interesting to us because it suggests that there are kind of two distinct mechanisms at play. And one mechanism might be, completed already, by the time a kid is adolescent. And that might be the impulsivity. And it is concurrent, right, with, for example, ADHD. You don't typically diagnose ADHD at age 14 or 16 or something. You diagnose it much earlier than that. So that means that the brain processes that underlie impulsivity, seem to be already established by the time the kid's 14, right? However, in addition, there seems to be a second, and this is, by the way, mainly in boys. There seems to be a second process going on, and this is more like a structural brain, like a brain developmental process, which doesn't necessarily have an immediate behavioral correlate that we could identify. And we also found it more in girls. And what we find here, that these brain changes between 14 and 19, that they are really also causing the increase in binge drinking. So you have one process that's sort of happening in a pre-adolescent phase, and there is another process that seems to be happening during adolescence, or at least, not stopped, or, still happening during adolescence. And both of these processes, in independent ways, influence increase in binge drinking. And if you sort of look at an interaction between the two, then you find, sort of an extra increase in binge drinking. So this was just, I thought that might be interesting for the, we were surprised. So the other, and this here is work by Rob Whelan and Hugh Garavan. Of course, this ties into the question, what does it take to predict binge drinking, right? As I told you before, we've been characterizing in the image ensemble, we've had really deep phenotyping. And what Rob and you did is they selected 115 binge drinkers at age 14, 150 controls that didn't take any alcohol or at either 14 or 16 years. And then as a sort of validation/replication sample, they looked at 121 future binge drinkers. So kids that didn't binge at 14 but did binge at 16. Now, here we go. Classification is very good, sort of to sort of identify at so then what they did, they used all the information that we have in the image ensample and used a regularized sort of a machine learning model to predict binge drinking either at the same age at 14 or in the future at 16. And using all these variables that we have available, the classification at age from 14 to 14 years was, very good, 91% correct. And actually, even the prediction for the future was not, great, but, very respectable. Now, what are the measures that are being taken? So here you can see it, and what role do those measures play? So these are, this is just a list of the measures, and I think what is immediately sort of obvious is that these things here, smoking, and even if you throw out smoking and look at the hierarchy, doesn't really change much. Romantic events, deviance events, family events, these are really sort of the best predictors for future binge drinking. Yeah? So that is important. It's important for two reasons. Firstly, it's important because these are measures you don't need an MRI for, right? So you can easily assess those measures. That's a good thing. And secondly, the second reason why it's important is it might predict well, but it really doesn't tell you anything about biology. So that's something we learned. But having said that, remember impulsiveness? So here, impulsivity, yeah? So that scores relatively high among the behavioral, among the predictors. Now, why am I showing you this model? Let me just check my next slide. I'm showing you this model because I think it bears, actually this bears really good immediate translational potential. And it's very simple. So what you do is you do a gold standard model, a full model, where you throw everything you've got at a defined number of individuals and try to predict, sort of an outcome of interest. Yeah? And then you go back and then you sort of, dissect it. You look at what are the individual components, how well do they predict? Yeah? And then you say, okay, personality and history together, is likely to predict just as well as the full model. And then you say, okay, so then let's make a questionnaire that looks at at personality and at history, yeah, and then we roll that out, right? And it's not going to cost 10,000 euros to make an assessment, it's going to cost 10 euros. And because it costs 10 euros, we can do it in schools, we can do it in a really public health relevant setting, while at the same time knowing that this questionnaire is calibrated against the best possible, so to speak, diagnostic methods. So I think that's important. Yeah, I'm typically, I run out of time, so what, I'm still, how am I? I'm still okay? Yeah, okay. Yeah. So remember, I was talking about my little linguistic problem, the English and the French, and how we bring together the behavioral, sort of the ways we organize behavioral data and the ways we organize neuroimaging data, and how we sort of pray for an overlap, which then more often than not does not manifest itself in the desired way. So we try to address that. And we try to address that using a method called sparse canonical correlation. And that's a very simple method also, conceptually quite, simple. What it does is you take all your neuroimaging data and you just, sort of dissect it into voxels, right? Simple. On the other hand, you take all your behavioral data, for example, an ICD-10, structured ICD-10 questionnaire, such as a SCID, or in our case, DABA, and you don't care about the diagnosis, you just look at the items. Yeah? And so then you have a long list. And then what this canonical correlation analysis does, it sort of identifies groups of voxels with groups of symptoms on the basis of the best correlation. Yeah? So you're deconstructing, if you will, your two languages, or the neuroimaging data and the behavioral data, and then you're sort of reorganizing them on the basis of how they best correlate. And Alex is my postdoc who is doing this. I mean, he's working like 5 days now and submitting this manuscript. This is after 7 months of revising. So he's, well, yeah, okay. I hope it will be done when I'm home. But anyway, so what Alex did is he used, again, from in this case, from age 19 individuals from the Imogen study. He used structural data, VBM data. He used, so gray matter volume. He used white matter connectivity. And he used resting state brain activity. And then he sort of integrated those into a full model. And then, this full model was then subjected to this canonical correlation analysis. And as I mentioned, the behavioral symptoms were the DABA, which is a structured ICD-10 questionnaire for adolescents. Yeah? And as a consequence then, what he now called is a multiple, sparse multiple, because there are multiple modalities here, sparse canonical correlation analysis. As a consequence, he identifies about, I think, 13 symptoms or so, which could be described as effective anxiety-based symptoms, but also with some thought disorder symptoms. And he links them, with particular, gray, features in frontomedial brain, gray matter volume, connectivity features, and also structural connectivity features and resting state connectivity. So the slide doesn't show it, but really the greatest, the strongest, so to speak, factor in this full model, and the only factor that was in itself statistically significant, was the structural, the gray matter volume. So white matter connectivity and resting state connectivity did not on their own contribute. And in fact, they didn't, contribute all that much to the variance in the first place. So we stuck essentially with the structural brain correlates. Right, and you can see here that in the test sample we got a correlation of 0.56, which is explaining it's like 20% or so, right, of the variance. But of course we had to replicate this in a test sample, and here it dropped. We're explaining about 5% of the variance. Still not great, but better than many of the other work that one sees. Yeah. And remember one thing also. So I think the task now for us is we can just systematically test, which are the modalities that contribute best to those symptom correlates, right? So we can look at genetics, we can look at epigenetics, we could even throw in environmental variables into the model, and then sort of step by step we can sort of optimize the combination that is necessary to explain the greatest possible amount of variance of behavioral symptom group, while at the same time maintaining biological interpretability. Now, these were also quite nice because the brain correlates that we identified did not only, they were already present at age 14, where the behavioral symptoms were not present. So with these brain correlates, we could already predict at age 14 the emanation of behavioral symptoms which are only manifest at age 19. That's an important one. And the other thing we could, and this is again the transdiagnostic part, which refers to your talk, and also so what we could do is if we identified, I didn't say that, sorry, we identified two brain correlates, one the frontal, one whose biggest component was in the frontal-medial cortex, and the other one whose biggest component was limbic and temporal. And if we use those correlates as regions of interest, as masks for brain, and looked at case control differences, we realized that we could predict, to quite a nice degree here, depression on the basis of this limbic temporal part. We could, to a lesser degree, predict ADHD, and then, down here, schizophrenia. We could not predict bipolar, neither with the limbic temporal nor with the frontal. So the reason we think we were not able to predict bipolar is for two reasons. The first one, the symptoms are of course, changing. That's the nature of the illness. So you might not get, so to speak, consistency symptomatology-wise, and we didn't have the possibility to, sort of select a sufficiently large number of a large group of bipolar patients that were really depressed only or something like that. And the second reason is bipolar patients often get treated with lithium, and lithium in turn is known to increase brain, sort of alter brain structure, increase gray matter volume, and mind you, these patients are adult. But the take-home message from this is, okay, so we can predict quite nicely using between 1% and 2% of the overall brain volume. We can predict, at least for example, depression just as well as using the entire brain. Yeah, number one. Number two is, okay, yes, we can predict, but it's not specific to diagnoses. Right? So it goes across diagnosis. There is a bit of a weighting here between here and here, but at the end of the day, these are transdiagnostic features, which is very much in, sort of in accordance with our expectations. As remember, reward processing, dysfunctional reward processing can manifest itself in very many different ways, and as such, contribute to very many different diagnoses. Right. I'm just going to skip this because I'm still afraid that I'm running out of time. Now, environment. So, about 50% of the variance, give or take, is explained by mental disorders, is explained by genetic factors. So the rest is environmental factors. Environment is really complex. And the way we typically measure environment is kind of very, very limited, right? We're looking at psychosocial stress, we're looking at trauma, maybe, some sort of birth-associated situations, perhaps parental smoking, that kind of thing. But if you're looking at this, you can see, that there are so many more facets to environment than we are sort of used to looking at, that it's a little bit daunting. Yeah? So that's one important aspect. The other thing is, if you're then looking at if you're assuming that mental disorders are significantly caused by environmental influence, or well, influenced at least, if not caused by environmental influences, and then you look here at the just different relative distribution, yeah, of psychiatric disorders in developed countries, in China and India and other developing countries, you see that the pattern is different. So as a consequence, one possible assumption is, yes, there are going to be ethnic differences between the Chinese and the Europeans, but it's probably also because the environment is different. So that means, if we're going back to the personalized, I've got the right slide, to the sort of precision medicine approach, yeah. So when we say that we want to identify disease treatment and preventions that target etiological mechanisms and take account individual variability in genes and so forth, we must also take into account environment and lifestyle. And again, most people do not live in Europe, so I think it's a good idea to go global about this in order to at least potentially benefit the greatest number possible. So one example then that we had along these lines is the CIVIDA study, where we're looking at the effect of environmental influences on brain development and behavior in India and across, 6 Indian sites, 10,000 individuals we are aiming to study. We have a bit over 5,000 now, and about 15% of those will be scanned. It is an accelerated longitudinal design, so spanning the age from 8 to 23 years, and we are just sort of hoping to sort of identify trajectories that sort of then cross, the later childhood and adolescence on the basis of these 10,000, and then look how the various environmental influences that in this particular project range from neurotoxins all the way to social-cultural factors, how they influence brain development and behavior. Now and in very preliminary, in very preliminary studies, you can see this is comparing, CIVIDA and Imogen, they were characterized as similar as possible. And you can see here, the blue ones are the CIVIDA and the green ones are the Imogen, and you can see that by and large, the Indians are much better people than the Europeans. Yeah, externalizing behavior is less pronounced in India than it would be in Europe. Internalizing behavior here is also, there is a different distribution in the two cultures and continents, and similarly with pro-social behavior. So already you're seeing that if you're comparing aged, matched, and population-based individuals from India and Europe, your personality profile is different. And that is one first indication for us, that it is of merit to study the effect in the different countries. Yeah, I'm racing practically. And this here is, so because this study was going on, we have contacts with China, and we noticed that in China that they're starting a lot of cohorts. So as a consequence, we talked to them and said, look, why don't you sort of take at least an assessment light that is similar to Imogen and CIVIDA, and actually also the ABCD study, and apply it to your cohorts. And then, and they agreed to that. And then in order for us to sort of be able to actually compare this, we're in the process of installing like a informatics and neuroimaging suite, an automatic federated data analysis to enable the simultaneous data access. So somebody can, sort of make a study, say, for example, from Lille or Nancy, and and then sort of punch in a question, select a particular statistical analysis method, and then run this analysis method in Europe, in China, in India, and in the UK and in the US. And they don't get any of the data that are gathered there, but they do get the results. Yeah, so that's a good combination between meta-analysis and deep phenotyping. And the last thing, and this is sort of the icing on the cake, you're having all these different countries, and you're having all these potentially different environments, and you certainly have very different quality of environmental data and access to environmental data in China, India, and Europe. Yeah, so we needed something that would bring it together, right? Sort of a common denominator on how we could measure environment. And we're interested in urbanization, and if you're looking here, these are satellite pictures of the development of urbanization in Shanghai between 84, and so we have longitudinal data, 84 to 2016. So we figured let's use satellite data and see what kind of environmental measures we can derive from these satellite data and how we can relate them to brain and behavior. And as a starting point, simply because it's accessible, we used UK Biobank data. And what you can see here is correlation of satellite features of urbanization and behavioral symptoms. Just the take-home message, yes, there are correlations. Here is alcohol. So alcohol is apparently correlated with night lights. Night lights, in turn, is a measure of urbanization, right? So and it's a positive correlation. So the more urban, the more alcohol, just as an example. And yes, there are also effects on relations of satellite pictures and brain. What we find here, and this is still very preliminary, we're looking at ambient temperature, and we find that ambient temperature is correlated with cerebellar volume. That is, I think, I don't know, this is very, it sort of triggers fantastic thoughts. It's quite interesting. But again, it's a very strong correlation. It's 10 to the minus double digit. It's really strong. Yeah, all right. With this, I'll leave you to your own thoughts. And I'd just like to thank everybody who was involved in the study and thanking you for listening. Thank you.